Now that we've talked about meiosis, we can talk a little bit more about the life cycles and a little bit more in depth. And again, when you go in Botany 2, you are going to talk a lot more about life cycles. So this is kind of a precursor, but I wanted to do it right after you talked about meiosis, because then now maybe you understand that meiosis produces haploid cells. So within the anther of the male or the stamen part of the flower, you have 4 embryo sacs. And in each one of those, you're going to have microspores, or actually you're going to have, these are microsporophylls. They're microsporangia, and they're going to produce individual microspores. And those microspores, inside each one of them, they are going to undergo the meiosis process and produce 4 microspores. So this is a, microsporangia. This is the microspores, 4 separate ones after meiosis occurs. They could both be called microspores, so that's a little confusing. But anyway, so you have the 4 microspores. Now these are haploid. And each one of those is going to become a pollen grain. So what's going to happen, remember that in order to be a gametophyte, you have to be a multicellular multicellular body. So this is how plants still have gametophytes. So each one of those microspores is going to do mitosis and produce 2 cells. One is called a generative cell, and the other one is called the nucleus of the tube cell. And that generative cell is going to do mitosis again and make 2 sperm cells. So within each pollen grain, I have 3 separate haploid cells, or nuclei actually, within that pollen grain. All right, so within this pollen sac, I have many of these microspores, or that is the mother microspores, that are going to produce the haploid microspores that do this, okay? Now, in the female part, of course, this is in the carpel, right? So I have in the carpel, I'm going to have an ovule, and each ovule is more close up, looks like this. So the whole thing is the ovule, and inside the ovule, or it's surrounded by integuments. There's two of them. By the way, in gymnosperms, they would only have 1, so that's why it's called unprotected seeds. One of these is going to be the seed coat later. And then anyway, the megaspore is the mother cell, and that is going to undergo meiosis and create 4 haploid cells, okay? And then those are going to do mitosis and create another cell each, so that makes 8 cells total, okay? So meiosis created the 4, and the mitosis created 2 of each of those, so now I have 8 cells. 3 of those cells migrate to one end, 3 migrate to the other end, and 2 migrate to the middle. These 2 that migrate to the middle are called polar nuclei, okay? And these guys down here are going to be the ones that get fertilized. So let's look at that a little more closely. So if I look then at this is the mother cell, so there they are. I'm going to have, this is the ovule again, and these are the 3 cells at the top. The antipodals is what they're called, the polar nuclei, and then the 3 at the bottom. And what happens is the sperm cell, which will grow down the pollen tube. So what happens is pollen lands on here. The tube cell grows down into the carpel, and it basically is hollow and allows the sperm cells to come through it and come to the what this is called the micropyle, and it'll come in there and fertilize one of these. The other 2 get reabsorbed, and it fertilizes these 2 together. So this becomes a 3-end structure called the endosperm, and this becomes a 2-end structure, which is going to become the little zygote. All right, so that's the thing that is actually going to become the baby plant. So this is just another version of the life cycle, kind of the same idea Actually, I think I'll skip that because that other one was better. So let's look at this. So after I have a baby plant, okay, inside of the actual seed, I'm going to end up having like a complete baby plant within that. And what happens first is that that 2N cell is going to divide, and then it's going to divide again, and then it kind of ends up making these different structures. So the very first part is what they call the suspensor. That's kind of the part that attaches to the outside of the integuments. And then these guys are going to start developing and creating the structures that are actually eventually going to become the complete baby plant. So these are kind of the gradations that occur. And what I will have at the end is I will have a root apical meristem, and I will have a shoot apical meristem, which would be up there. And then I'm going to have the cotyledons that are coming off of that. That is the, cotyledons, usually there's two. This one's kind of just showing you half of it. And then I have the central axis, which is the procambium, that eventually that's going to become the vascular system. I'm going to have the pericarp on the outside. That's kind of the harder part. And then I have the seed coat that is just inside of that. That's where the two integument parts are coming from. And those are going to be protective of the little baby plant now. And so that seed could land on the ground and germinate if it has the right conditions. All right, so after germination, if I'm a monocot, basically the seed can sometimes be pulled out of the ground. In other words, here's the seed and the shoot. Remember, in a monocot, I have one shoot that is going up, and it's going to pull that seed out of the ground and start growing up. So an onion is a typical kind of monocot that would do that. Or I could have, like in the corn, where I have that actual shoot, which is called the coleoptile, just coming up from under the ground, leaving the seed behind, and then the fibrous root system forming and the shoot forming. And remember that monocots, the newest things are growing from the central or from the base, and we kind of talked about that before. And a typical eudicot, on the other hand, they can do the same thing. In other words, they can leave the seed in the ground and grow out, and that's both of these cases, or they can pull the seed out. This is like pulling the seed out with the cotyledons out, and then the new parts will form. All right, so in this case, what is happening is that the plant will start to grow its root system, and it creates this little, see if I can show you there, the hypocotyl, which is like the part that's before the actual plant, and then before the leaves and such. And then that part, as it continues to grow, will pull the cotyledons and the foliage leaves out, and they will start to form and then grow into the major part of the plant, and then the cotyledons will wither. The cotyledons are basically like nutritive structures, and they would be most likely formed from the endosperm. And then in this case, this is another way that could happen. You could have the hypocotyl pulling the whole seed out, and then the cotyledons would be, again, left behind and wither off, and the new leaves would start doing photosynthesis. And or you can actually have the seed in the ground, where both the shoot and the root axis start to grow up and down separately, leaving the seed there. Okay.